Okay, good morning, everybody. Thank you so much for coming. It is my pleasure to introduce Dr. Vivek Srinivasan. He is a leading expert in the fields of OCT of the eye, as well as diffuse optics of the brain. He did his doctoral training at MIT, followed by a postdoc at the Martino Center at Harvard, where our paths briefly crossed. And then he went on to a faculty position at UC Davis, and now he is associate professor at NYU's Grossman School of Medicine. And I'm so excited to have him here today and to hear all about the cutting edge stuff he's been doing. So please join me in welcoming Dr. Srinivasan.
Okay, thank you so much. And I hope everyone can hear me in the back. If not, just let me know. Feel free to stop me with questions if you like. If this can be interactive in this big room, like it to be so. So my work is in the field of biomedical optics and biophotonics. We have a lot of great faculty here in biophotonics. Aaron, Shuja, as well as Paco Robles, who I'm meeting with later. So it's a field that is well known to the campus, basically the combination of biology and optics. And I work kind of more on the medical imaging, medical side of things. And actually, this is really interesting to think about really how broad a role optics plays in medical imaging. If you take all of the optics and including like ophthalmology, endoscopy, and lump it all together, actually the market is larger than what we think of as conventional medical imaging, being radiology, MRI, and PET. So not to say that we're better or larger than them, just that optics is important and ubiquitous, even if it's not recognized as a field. And so one of the themes that kind of ties through my research is that in this field, we can use advances in areas like telecommunications. So these big economies of scale that have nothing to do with medical imaging, but can benefit medical imaging technologies. So in my class, I often ask students, how much do you think a 1,000 feet of single mode optical fiber costs? Any guesses? So about $200 from a Thorlabs website, and likely even cheaper if you buy it wholesale. So as Aaron mentioned, the work in my lab kind of spans several different areas. And so when we think about optics and light tissue interactions, it's typically divided into microscopy and macroscopy. No one says microscopy, they often say diffuse optics or near infrared imaging. And these two scales are really determined by how the light propagates through tissue. So for microscopy, we're typically looking at the light that travels straight through the tissue. So think of like a 2-photon microscope, where the light goes to the focus and comes back, or goes to the focus, excites fluorescence, which comes back. So this light travels through more or less without scattering. On the other hand, diffuse optics uses the multiply scattered light that penetrates much more deeply into tissue, but also with lower resolution. And so one of the advantages of my research, kind of working in different areas, I'm able to look at the same organ from several different perspectives. So this is an example of OCT angiography of the rat brain, showing the microvasculature with a lot of detail. I'll actually come back to this picture in a moment. And then also recently, as Aaron mentioned, we've also moved into applying some of the similar technologies or new technologies to look at human brain imaging through the intact scalp and skull. So this would fall under the regime of diffuse optics, where we're using multiply scattered light. So one gives us nice pretty pictures, the other kind of blurred images, but we can get those blurred images in human subjects, which is really, really great. So I'm going to do a little bit of a dance back and forth between the micro and macro. My talk is focused on the human brain, but I'm also going to intersperse, let's say, some pictures, microscopic images from our work in mice that can help shed light on some of the brain functions that we're looking at in humans. So my work, as was mentioned, falls into 3 different areas. We have a deep brain imaging side of our lab, where here's an example of imaging the cell bodies in the mouse brain through a relatively non-invasive preparation. This is in mouse. You can image all the cortical layers and even some subcortical layers. We have a great deal of our work in eye imaging, which I won't talk about today, but hopefully can touch on with some of you in conversations if you'd like to discuss. We're here using visible light OCT to make high resolution pictures of the retina. And finally, what the focus of my talk is applying some of these interferometric tools to image the human brain. Okay, so I'm going to talk a little bit about OCT. This is optical coherence tomography because it's a tool we use in our lab, but also it gives some ideas and foreshadows some of the later work I'll be presenting. So optical coherence tomography is basically a technique that was developed as the optical analog of ultrasound. So the idea is we take a pulse of light and we visualize backscattered or back reflected echoes of light. Now, because speed of light is very fast, we cannot actually do this directly. So we need to build what's known as an interferometer. The basic idea is we have a reference path and a sample. We interfere light on the detector. And here's a simple illustration. We can see we have, when we interfere two fields on the detector, we have sample field, reference field, and then a cross term between them. So that cross term is actually very important. In the area of OCT, we can make the reference field very large. So if the sample light is small, we can boost it up to a level that can be measured with a relatively inexpensive detector. The second aspect of this picture, which I want to highlight, is here I've shown an interferometer with a coherent light source, so that if I change the path length, I'll get a series of fringes. So coherent light is very much like this laser pointer that I'm using. And if you're on Zoom, you'll just have to imagine the laser pointer. Now, if I take that coherent light source and replace it with an incoherent light source, so incoherent light is very much like the room lights that are shining down from above, then actually I am able to resolve different depths in the sample. So here's an example of resolving depth in the sample with very fine spatial Z resolution by using incoherent light. And that occurs because light will only interfere when the paths are matched to within the coherence length. So OCT, you can focus a light beam on the sample. One of the great aspects of this field is if you look at a typical OCT system, it has fibers, it has filters, demodulators. So these are all fiber optic components, telecom components that have relatively inexpensive economies of scale behind them. And as a result, this technology, since its invention, is now widely used. So this is a $1.5 billion market as of 2021. It's likely even larger today. Many of you, if you go to an ophthalmologist or even an optometrist, it's likely you've had an OCT scan. See some nodding heads. Okay, good. So this is widely used and very useful. And I think the rest of my talk, I want to come back to this example in terms of how we can use some of these concepts to build a brain imaging device. Okay, but this talk is about the human brain, so let's get on to the interesting part. So why measure cerebral blood flow? So the first reason is that it's the brain is about 2% of the body weight, but consumes 15 to 20% of the energy demand. If it's perturbed in brain diseases, we'd really like to monitor this over days to weeks. So I have several examples here: traumatic brain injury, subarachnoid hemorrhage, and stroke. So you already have a great faculty member here, Aaron Buckley, who's already studying a lot of these conditions. So I don't need to make the argument for optics, but the basic idea is that our goal is not to do better than what an MRI can do or do better than what a CT can do. The goal is more to go places where these conventional imaging modalities cannot go. And in the case of, for instance, traumatic brain injury, which we're pursuing in our group, the goal is to monitor patients for weeks, days to weeks after an injury. Whereas with an imaging device, you'd have a simple snapshot. With optics, we can monitor for long time periods and tailor the treatment specifically to the patients. All right, so the second reason to measure cerebral blood flow is because it's a great reporter of brain activity. So this is a bold functional MRI you already have in Shella Kelholtz, faculty member who's really getting at the fundamental mechanisms of the Boole signal. But basically, it's a blood flow signal. And so by measuring blood flow, we can actually infer what's going on with neuronal activity. Okay, so there are a number of surrogates for brain monitoring, all of which are imperfect in some way. Transcranial Doppler, it measures velocity, perfusion, MR and CT we've talked about. And there's a number of other surrogate techniques which do not directly get at the cerebral blood flow that we'd like to measure. And so now, as I mentioned in my group, we use OCT to image the mouse brain. This is an example, of the rat brain with the skull and dura removed. So this is a relatively invasive preparation, but it gives us beautiful pictures of the microvasculature. You can see here the arteries, which arise from the great cerebral arteries that supply the superficial cortex, diving down into the tissue, the veins coming back up, and we kind of have this mess of capillaries, spaghetti, which is actually where the oxygen delivery to the tissue takes place. However, our goal is to measure the human brain. So if we think about the penetration depth of OCT, that ballistic light, it's really only going in a millimeter or 2 millimeters. Now looking at this far corner picture, we really want more penetration depth to get through the scalp and skull to reach the brain. So we have to use a different approach. This approach is known as diffuse optics or near-infrared spectroscopy. This is a great field. It's had great wide utility. It's perhaps because of its simplicity. So that all you need is a source and detector, and you can measure the light that travels between the source and detector. So very simple, very useful, but with that simplicity comes a cost. And I really wanted to highlight two issues. So the first is the source and detector are separated. So because the light has to travel between the source and detector, the spatial resolution is perhaps compromised. We want to go to larger separations to get deeper. The second is you can see that light has to travel through the superficial tissues to reach the brain. Because of that, the scalp and skull have vessels. There's blood flow in the scalp and skull. That blood flow fluctuates according to the vagaries of systemic physiology, which might have nothing to do with the brain. And so this is a contamination on top of the brain signal that we'd like to measure. And I would say this issue of scalp contamination is not often mentioned when we talk about this, but when you think about it as people who do diffuse optics, like this is the problem in the field. So if we could solve this problem, or at least if not solve it, mitigate it, we could really extend the range of applicability of these technologies. One other approach, which I'd like to mention, is also a diffuse optical approach, is called the DCS. This is developed at UPenn originally. The basic idea here is I take laser light, very similar to this laser pointer, shine it on the brain, and observe the light fluctuations that occur when the light scatters off of moving red blood cells. And so basically, slow fluctuation means less flow, higher fluctuations mean more flow. We can quantify this by an autocorrelation function that tells us how fast the blood flow is moving. There's a number of models that allow us to do that. And we come up with a blood flow index. And so this is a basis for a number of work. This is a growing field. Aaron has made great strides in pushing this technology into the clinic and doing it in patients, which is very impressive. And so this is already an area that has had impact. One of the things I like to think about, so again, going back to the microscale, we can also look at the vasculature from a standpoint of a diffuse optic system. So this is actually an example of the mouse. This is kind of an en face image of the scalp, of the skull and the brain. And I'm using a OCT angiography, so I've enhanced contrast from moving red blood cells. But basically, we can see the superficial vasculature is kind of a net or a meshwork on top of the deep brain vasculature that we'd like to measure. Okay, and so as I mentioned, the superficial vasculature, it's a noise. So as neuroscientists, I assume we're not interested in the scalp and skull. We're interested in the brain. So the real problem in the field is how to deal with these. One of the great advantages of DCS signal is that it's less sensitive to the scalp. So here I'm showing a plot of the brain to scalp sensitivity as a function of the source collector difference. So with the conventional near signals, which are based on absorption, as shown in the black line, we can improve a little bit by going to high source detector separation, but we really never get above like 40% brain to scalp sensitivity ratio, which means we're actually more sensitive to scalp than the brain. With the flow signal, with this fluctuation signal, actually we do much better. So the red line shows the DCS signal. We can see that actually at the larger source collector separations, we can go get even higher brain specificity, and in fact, make our measurements more sensitive to the brain than to the scalp. However, current DCS measurements, as for the reasons I'll mention, are actually limited to short SC separations. We'd really like to go even further. So how do we do this? One example of why this is a key question, this is taken from a DCS study that was done in patients, I think in the neurointensive care unit. These are measurements of cerebral perfusion pressure, intracranial pressure at the same time. And the goal was to look at autoregulation. So if we look at one epoch, we can see that as the arterial blood pressure changes, the cerebral blood flow is more or less constant. This is a marker of intact CBF autoregulation. If we look at another epoch, we have now a case where cerebral blood flow changes with mean arterial blood pressure. And so the question is this impaired CBF autoregulation, or is this a simple contamination of the superficial tissue onto the deep? Okay, so this is a key question we'd like to solve. So with DCS, as I mentioned before, we're kind of limited in the source detector separation. You can get more detectors, but these are relatively expensive detectors and difficult to scale, although some groups have gone this direction. Our idea, which we've first tested about 5 years ago now, is to use an interferometer to make us more sensitive to the deeper light. So we add a reference arm to our near setup, just like we do have an OCT. So if we have a reference arm, then the sample arm becomes the object or the brain. And I will argue this is a result. The result is a new class of methods for making non-invasive in vivo measurements of the human brain that are both more informative and less expensive than other techniques. Again, we get this benefit. Now we're multiplying a weak sample light field by a strong reference field so we can boost up that signal. And we can use less expensive detectors. So I will show progressively moving towards cheaper detectors, CMOS detectors, even eventually detectors that are similar to those used in your cell phone. And now if we can make each of those pixels as sensitive as a photon counting detector, we can now scale the measurement accordingly. And then the second, which is kind of interesting, is we can use some of the low coherence concepts we borrowed from OCT to give us time of flight resolution, which will allow us to distinguish layers more directly. Okay, so to the actual setup. So this is the basic idea. We have a reference in a sample arm. We interfere the light onto a CMOS camera chip. For the first studies, we used a fairly expensive camera chip. So this is around $5,000. It's a line scan CMOS camera, but with a very high frame rate. We need a frame rate in the range of hundreds of kilohertz in order to sample those very fast field fluctuations from the brain blood flow. Okay, so this is a postdoc, Wenjun Zhou, in the lab. This is the first system he developed. I hope the movie plays. Okay, this was obviously done at Davis, as you can see. One of the interesting aspects of this invention is we can do it with the light on. So when we apply a very strong reference field to boost up the sample signal, we're actually less sensitive to ambient light. So we actually don't worry too much about stray light affecting the measurements, which is not the case with photon counting. Okay, so this is during the pandemic. As you can see, blood flow index is shown up here. This is a pulsatile blood flow averaged over time. We can get the heart rate from that, et cetera. Okay, so now to the limitation of the field. So with this technique, we're actually able to push the source detector separation. Again, each of those pixels is now acting more or less like a photon counting detector. And we have many pixels, in this case, 500. Okay, so we can now push beyond 3 centimeters to around 4 centimeter source detector separation shown at the bottom. We still get reasonably good pulsatile blood flow. And I'll mention, even we can push it up to 5 centimeter source detector separation, although we do run into problems with sampling of the camera. So we decided not to go up that high in practice. And importantly for us, as we increase the source detector separation, we get more sensitivity to the brain. I didn't mention it before, but the brain blood flow is about 6 times higher than the scalp blood flow. So as we get more sensitivity to the high flow rate region, we should expect the blood flow index to go up, and that's actually what we see. Okay, so we have a new technique. Now, how do we validate this? Difficult to validate. As I mentioned, there's no real gold standard for measuring cerebral blood flow continuously, but we chose a test of cerebrovascular reactivity using a hypercapnic challenge, so a carbon dioxide challenge. It dilates the blood vessels, increases cerebral blood flow. Here's the protocol. So here, just skipping ahead, here's the final results. So we have end-tidal CO2 going up, and we see a corresponding increase in cerebral blood flow. Every time we have a challenge with CO2, we can see blood flow increase. Now, this is in human subjects, I should mention. Okay, so just blood flow increases during hypercapnia, but other aspects of brain measurables change as well. Now, going back to a mouse model, so this is now just showing kind of what's happening to the brain vasculature zoomed in during hypercapnia. We have an artery coming in red. I should mention this is using visible light OCT with this oxygen saturation. We have an artery coming in red and the vein draining the cortex in blue. When we apply carbon dioxide, let's neglect changes in oxygen metabolism if they occur for a moment. Now with hypercapnia, we have an influx of fresh cerebral blood flow. That blood flow will increase the oxygenation. So the arterial oxygenation doesn't change too much. But if we look on the venous side, this vein here, which was previously in blue, indicating a low oxygenation, is now in red. And arterial, I should mention, also dilates. This is the reason for the blood flow, increased blood flow coming in.
I have a question.
Yep, go ahead.
Is there shunting between the arteries and veins? Is that why that happens? Or is it just
So oxygen extraction decreases because blood flow goes up and metabolism stays the same.
but at all levels? Or I guess oxygen extraction. Are there arterioles that go straight to venules, or is it like?
That is a good question. I'm going to say I mean, obviously there are exceptions, but that is not the standard route of.
You can answer that question here. So
Yeah. So I mean, I think this is a real question of oxygen extraction decreasing. So if there's more flow going in, I think the majority of the change in veins is due to just decreased oxygen extraction. So oxygenation goes up. I'm just trying to think in the mouse if there's shunting. I'm going to say probably not, but someone might correct me if I'm wrong. Okay. So we have absorption change that goes along with this blood flow change that we can also measure. And so this is an example with our system. We can also measure like the conventional NIRS signals. And we can measure an absorption change, which is kind of a convolution of the oxygenation as well as the blood flow changing. And we can plot that as well. So we see here, actually, if you kind of squint a little bit, you can see there's a absorption change in red and a blood flow change in black. The absorption change kind of lags the blood flow change by a little bit, which you would expect from a kind of a washout effect. But now we can do this across many subjects. We can plot the end-tidal CO2 versus the different potential biomarkers. So we have end-tidal CO2 on the x-axis, blood flow change versus the absorption change. And we can see that blood flow is actually much more tightly coupled with our hypercapnic challenge. If you look in the cerebral blood flow literature, you expect about a 4 to 6% change in blood flow per every millimeter of mercury end-tidal CO2 change. We do see a fairly linear relationship, which is a good check, sanity check on our measurements. Now, if you look at the absorption, this is now a near signal, what conventional mirrors measures. We don't see as tight a coupling. This could be because we're not measuring directly oxygenation or blood volume, but kind of some convolution of the two. Another reason is that near signals has more scalp contamination than brain contamination, as I mentioned. So the fact that the scalp is kind of doing its own thing during these measurements may reduce the tightness of this correlation. Okay, so now the second validation which we can do is to look at now a brain activity measurement. This is a mental arithmetic task. Again, I'm showing the blood flow index measurements on the left and the absorption measurements on the right. So this is the BFI, kind of our CBF measurement, and our absorption measurement on the right, which is more like a hemoglobin kind of measurement. And we see that during this mental arithmetic task, we see a very clear, I would say very clear from my point of view, we see a blood flow index increase on virtually every trial. Whereas if we look at absorption, we see the story is a bit more complicated. So again, this is a good validation, not a perfect validation, but a good sanity check that we're able to do some of these measurements in human subjects. And I should mention this was at 3.5 centimeter source detector separation. Okay, so just now going back to the micro scale, just because I want to keep people awake with some nice images. So this is now an example of what's happening to the brain vasculature during an activation. This is now in the mouse during 4-paw activation. Let's not worry too much about that. The main thing I want people to see is that during the stimulus, which is from 0 to 2 seconds, you can see this vessel along the top kind of increase in diameter. There we go, it happened. Okay, so this is kind of thinking about at the micro scale, what's driving these signals that we're measuring? Well, it's really neuronal activity causing arterial dilation that's creating this influx in cerebral blood flow. Okay, and then we can also do hair monitoring. This is, again, like one of the things that people in the field don't talk about too much. So I will say, like in regions with hair, this is an example of motor cortex. We see a difference in size between contralateral and ipsilateral finger tapping experiments. This is still only a 2.5 centimeter source detector separation. So the technology still has a ways to go. But on the other hand, we can monitor in brain regions with hair, which I think is a good advance. So it's both motivating, but also saying there further improvements are needed. So to get those further improvements, we next asked the question. So we started with kind of a high-end camera where we're sampling these very fast field fluctuations arising from blood flow. Is there somehow a way we can take what is an existing economy of scale from CMOS camera technology and use one of those cameras to make our cerebral blood flow measurements? So here's just showing the growth in CMOS cameras. I would guess there's at least 2 times as many CMOS cameras in this room as there are people, possibly more. So this is a widely used device. So the answer turned out to be yes. The same postdoc who did the previous study, he discovered that if I vary the exposure time of the camera, I can actually sample the autocorrelation function in slightly a different way. So I'm not actually measuring the autocorrelation function directly, I'm measuring a weighted integral of the autocorrelation over the exposure time. And so by varying the exposure time, I can get information about how that autocorrelation function is changing. And so he's able to do these measurements. This is now with a 2D camera with megapixels, right? And he did 2 exposure times. One of the big advantages of this approach is that now with a 2D camera, you can now image different sources and different detectors or detector positions onto the camera to take advantage of the 2-dimensional array. So we looked at a long and a short source detector separation. And we measured, this is an example of measuring the blood flow index during breath holding. At the short SD separation, this is 1 centimeter. This is pointing out that we get a lot of signal from the scalp, particularly during a breath hold. And he showed At 3 centimeters, he's measuring brain and scalp, and he did some regression shown on the lower right in black to just isolate out the brain signal. Okay, and so with this approach, we can now say we are able to get a 4 order of magnitude potential reduction in the cost, in the performance to cost. Okay, so on the y-axis here, we have cost. We have an MR scanner. I actually don't know how much one costs. I just assumed it's $1 million. Sounds really expensive. It probably, yeah, exactly. Okay, so that's kind of a point of reference. And then we look at the different technologies. So with any of these optical technologies, we can always buy more detectors, right? And so the name of the game is to really make the detector itself less expensive. With the DCS technology, which again is the gold standard, been incredibly useful already, it is difficult to scale. And so if you just buy more APDs, this is how the technology is going to scale. So around the place where you get, I don't know, like maybe 1,000 detectors or 1,000 channels, you're around the cost of an MR scanner. All right. So with some of these new technologies now, we're using pixels instead of detectors. We can reduce the scaling. So it's still expensive, right, once you scale, but you can see now we can get, with our previous approach, around 10,000 channels before we get to the cost of an MR scanner. And now with the multi-exposure approach, we can even get to 1 million channels and still be less expensive than an MR scanner. And to me, as an engineer, this is really exciting. So whenever you've improved the cost and scalability, you can now ask the question, okay, so now I can make a device that is relatively cheap, maybe does something similar to DCS, but can be used in settings outside of a hospital or a medical environment. Okay, I'll talk about that more in a little bit. And at the same time, I can make a high-end device that might perform better, might measure more brain regions, or measure at larger source detector separation. So I will say, although we haven't solved the problem of scalp, I think we've improved the situation considerably. Okay, so taking a little bit of a different direction now. So the next question I wanted to ask is, now we have this kind of interferometer setup. Can we get other kinds of information from the interferometer? And here, one of the useful dimensions of information that has been explored in the field is time of flight resolution. The idea being that the longer times of flight will travel proportionally more through the brain and less through the scalp. And so by measuring time of flight, we can distinguish some of those signals. So here we don't actually have a pulse laser. We have a continuous laser. And we found that by using a very similar approach as is used in OCT, we tune the laser in time and change its wavelength. We can actually encode different depths as different wavelengths, different frequencies of the optical signal. So by tuning the laser, we can generate an interference pattern. So the scalp interference pattern will have some frequency. And then the brain interference pattern will have another frequency. And then by reconstructing through a Fourier transform, we can obtain a time of flight resolution, or TPSF, temporal point spread function, which characterizes the time response of the tissue. This is now one of the advantages of this approach is we can now get time domain information in terms of a time resolved measurement of the tissue. But at each time of flight, we can also get like a DCS-like measurement. So at each time of flight, we can also measure the light fluctuations of the light that travels through tissue with that particular time of flight. And I should mention there are other approaches in the field, such as time domain DCS, which try to do similar things. Now, some of you, I think maybe a few of you who have some background in optics might say, hey, this is just swept source OCT. Actually, it's true. It's very similar to swept source OCT, but we should think about the time scale of these measurements. So for NIRS measurement, we're really measuring up to nanosecond time scales. With OCT, we typically only measure up to around a 10 picosecond time scale. So the time scale is much broader, and so the depth of penetration that light has will be much greater, allowing us to travel to the brain. Okay, so now we take this time of flight resolved measurement. We can now look at different media. Some relatively unexciting media to look at. What one model in the field is intralipid. We can see that the decay rate increases linearly with time of flight. This is expected based on a theory. Look at the mouse brain too. The mouse brain, the decay rate or the autocorrelation decay rate also increases linearly with time of flight. This is, again, expected for a uniform homogeneous medium. As it turns out, the superficial tissues in the mouse are relatively thin compared to the brain. Now, applying this to the human head, the first thing we notice is that now the decay rate versus time of flight has a relatively non-linear characteristic. Actually, I would say it's a triphasic characteristic where the early times of flight have one slope, then it appears to decrease, or the slope decreases. And then at the later times of flight where we're sensitive to the brain, the slope increases. And so we puzzled over this for a little bit. What would possibly give us this kind of behavior? We looked through the literature. It's turned out that people have argued or debated the merits of different models for modeling the extracerebral tissues in diffuse optics. There's a 2-layer model which says the scalp and the skull essentially have the same blood flow. And there's a 3-layer model which segregates the scalp and the skull, with the scalp having a higher blood flow than the skull. We fit this models to both of these models to our experiments. And we found that the 3-layer model gets a remarkably good fit to the data, whereas the 2-layer model does not. So this says, at least to us, that the brain, scalp, and skull is best modeled by a 3-layer system as opposed to a 2-layer system. And I think this is very important, even for the conventional techniques that seek to get quantitative information from light measurements to know exactly how to model the superficial tissues. Okay, so the last idea that I wanted to present, which is building on this again, is so I've shown a way to obtain massive parallelization of the measurements using CMOS pixels. I showed a way to get time of flight information. Now, is there a way we can kind of combine these two approaches together into one single system, get both the time of flight information as well as the parallel detection? So to do this approach, we went back again to the basics of OCT. So the idea is we want to create a low coherence light source. But if you think about the timescales involved, you actually need coherence line width of around a few gigahertz. It turns out these sources don't really exist. So we had to make one. And the way we made it was kind of interesting. So we took a narrow line width laser. We tuned the wavelength very fast in time. We said, okay, if we tune the wavelength very rapidly on the order of megahertz in time, we generate what looks very much like a low coherence source. So there's a power spectrum shown on the right. Now that power spectrum will have a coherence function as shown here. Okay, so very similar to how OCT uses low coherence light to generate images. Here we're using a effectively low coherence light source to filter out photons that travel deep into the tissue. Go through some of the math. I think that's probably less important. I mean, the basic idea here is if you look in panel D, I start with a distribution of times of flight shown in blue. I apply a time of flight filter shown in the black dotted line, and the filter time of flight distribution is shifted to the right in time of flight significantly. So that means that I'm actually getting deeper photons by applying this time of flight filter. Okay, I'll skip over that a bit. Okay, so I'll mention some people may have heard of time domain years or time of flight years or frequency domain years. It's actually kind of a different approach. I would say it's maybe even a different class of near approaches that uses a variable time of flight filter to get time of flight information. But again, we get to the problem of how we validate this. So in addition to doing all the other validations that I mentioned, I wanted to highlight something, a pressure modulation experiment. Here we basically say, okay, we know our scalp signal is a confound. So if we squeeze on the scalp and we're getting a lot of blood flow signal from the scalp, we reduce blood flow in the scalp, that blood flow signal should go down. If we're getting less of the scalp signal, that blood flow signal should go down less. And so here we ask the question, can we measure the reduction in superficial contamination from the scalp by basically just squeezing on it? Okay, so we took a pressure modulation experiment. We did an approach where we turned the time of flight filter on and off very rapidly in time. So we kind of shifted from measuring what's CW, which is continuous wave, to time of flight filtered, which is deep photons. So we kind of did superficial photons, then deep, then superficial, then deep, switching back and forth every second. And so we're able to get a curve like this during a pressure modulation experiment. So again, I'm squeezing on the scalp, right? So this is a 1 centimeter source detector separation. So the blue line shows the kind of conventional measurement that when I squeeze on the scalp, I reduce blood flow to almost 20% of its value, which says really most of that signal that I'm measuring is coming from superficial tissue. And if I turn on my time of flight filter, this is again the same experiment because I'm switching back and forth. I measure a significantly reduced decrease. So in other words, I maintain that blood flow level during the scalp occlusion. So we can see other features here like the release after occlusion, you have fresh blood flow coming in. If you're just squeezing on the scalp, you presumably don't occlude the brain unless you squeeze really hard. So we don't expect the brain to have any response. So any increase in blood flow after the occlusion would be presumably due to the response to the scalp. So we can see that the scalp response is large with the continuous wave or superficial photons, but it's reduced with the deeper photons. And we can do these measurements across subjects and show we have a significantly reduced perturbation caused by the scalp. Now, one of the other great advantages of going to low source detector separation and using time of flight filtering is we can improve the spatial resolution of the measurements. The last thing I want to show is this concept of high density forehead mapping. Now we're down to about 1 centimeter resolution at the brain. So for optics, like this is pretty good. I know that the MRI people may scoff at this, but again, we're not trying to do what MRI does. Okay, we're trying to do it in a more cost-effective way. We can do imaging of these different parameters via the human forehead. Actually, what's interesting is this light intensity ratio. If we compare continuous wave and time of flight filtered light intensity, we actually get a very symmetric kind of a distribution, which I find to be kind of interesting, symmetric across the two hemispheres, which says there's some kind of a structural symmetry between the measurements. I think I'm getting something from somebody, huh? Yeah. Okay, so future directions of this approach. So we've shown we can improve this scaling of the blood flow measurements now. And of course, we're interested in neurointensive care and rehabilitation. We have a project, particular project with a traumatic brain injury, but there are certainly other applications that are very interesting. But now we have a scalable technology. So there are other kinds of questions we want to ask, or can we start to monitor this, outside of a hospital setting, for instance, in the doctor's office? There's a lot of questions about cerebrovascular health and Alzheimer's disease or other vascular dementias. Some of these tests of reactivity could potentially be done with a less expensive instrument. Maybe you don't need quantitative measurements. Maybe you just look at changes. And then even further, can we do this monitoring at home? So with a cell phone camera sensor, it's feasible to start thinking about can we start to measure cerebral blood flow even at home or even outside of a doctor's office. And then can we tie the loop back with preventative medicine? So can we use some of these data to develop preventative approaches to stave off some of these debilitating diseases? So in summary, does interferometry help diffuse optics? So I've given you kind of a positive viewpoint on this. I believe it should improve the brain to scalp sensitivity of blood flow measurements, particularly in cases where we're photon starved. I think it will help. We get some interesting ways of getting time of flight information, which is interesting for just understanding the extracerebral tissues and how they contribute to blood flow. And another question which has come up quite a bit is, can this technology or its variants compete with high throughput continuous wave nears for functional studies and brain-computer interfaces? And I think here the answer is possibly, but it will require further advances. On the other hand, we are making advances every year, so I am optimistic that it could still compete with continuous wave nears in terms of scalability. More broadly, biomedical optics, this field spans from macro to micro. I hope I've given some sense of the flavor for the different techniques that can be used. It aids both basic science discovery as well as clinical diagnosis. And one of the keys from my research is to think about how we can use larger economies of scale from communications or consumer electronics to advance some of these medical device ideas. And there are numerous unsolved and challenging problems for physicists and bioengineers. So lastly, I'd like to thank my research group and funding sources, and I look forward to questions. Thank you.
Hey, Vivek. Very nice talk. We're going to be meeting later, but I think there's a question that I think is worth putting up here with the entire room. Just getting to the weeds a little bit in terms of the signal that you're extracting with your interferometric approach. Are you using only the intensity fluctuations over time? Are you bringing in phase differences? What information do you get from each one of those different sources? And if you're not, what opportunities do you think there'll be by combining both the intensity and the phase fluctuations that you get in your interferometric signal?
Yeah, so if there's something called a Siegert relationship that relates the intensity to intensity to field fluctuations. So if the Siegert relationship holds, we're not really getting anything fundamentally new. Although there are interesting regimes where the Siegert relationship does not hold, where there's less light scattering, maybe few scattering events. I will say generally the signal to noise ratio is better for the field measurements in terms of the photon efficiency, but field measurements are sensitive to phase noise. So that is a potential issue. And so we do expect that these measurements, and we've seen it, that these measurements are a bit more sensitive to like stomping on the ground than a DCS measurement.
So it's all intensity based then?
I'm sorry, we are using the field. We have no choice.
Okay.
So we are sensitive to phase noise.
Got it.
Yeah.
Yeah.
So in terms of like patient motion, if the patient's moving, I think that the motion is similar. And that's also what we've seen in the collaboration with Arjun's group. But if the nurse comes in and stomps on the ground, then you're probably going to see more noise in our system than in a DCS system.
So you're saying if like the room vibrates.
Yes. And that has to do more with how we build our interferometer, and that it could be solvable with engineering approaches. We're just giving you, I'm giving you my observation based on what we've seen so far.
That's really interesting, because that's kind of tricky to know when it like at least when a patient moves, you can know oh the patient was moving, but it might be hard to notice vibrations of the bed that
Yeah. So I mean, it's something. Okay, so
right.
So okay, here's the way I'll put it. So right. So you could make the same criticism of OCT. That's also an interferometric system, yet it's used everywhere.
Yeah. So how do they?
But so that's the intensity signal that they're detecting for their signal.
But they also use phase information.
For Doppler. And
Yeah.
like that.
Yeah.
But I wonder if you can extract it from or get different information from the phase and the intensity of the OCT signal. And sure, they might be related, but then you might go off your SNR.
Yeah, I mean, we can definitely talk about that later. Yeah.
The mic is not working, so if anyone has any questions, just feel free to raise your hand.
Like current applications, other than just put it on mobile phone, how do you see it growing?
Yeah, that's a good question. I don't think I said put it on the mobile phone. I don't know. It seems like everyone wants to put everything on your mobile phone now. Yeah, this may not fit on your mobile phone, although it could, certainly process data, and we use a sensor like that. I mean, the idea was to use the economy of scale that's creating your mobile phone to make a device. Okay, so I mean, I think the idea is really to be rigorous about this and validate it in clinical settings first. That's the academic in me kind of speaking, but I think for a lot of these applications, I mean, if you're talking about commercial applications, it's tricky. I mean, companies have been down this road before of measuring cerebral blood flow, and it's not been a good road to go down. But for me, I'm excited about just the scientific implications of the technology. And I think when there's a real validation and acceptance, I think those other approaches, other applications, right, like putting them in other places, other settings will open up. But I think right now, I think basically what Aaron is doing is the way to go about it. Yeah.
What do you see as the ideal type of validation? Like if you were to have an experiment and you see an agreement with how would you say this is good?
Okay, that's a good question. So, I mean, I think there are a number of sanity checks that I would do. We've done some of them. I think transcranial Doppler is another good sanity check. I mean, I think if you ask what's the gold standard, it's like perfusion MR or CT. It's a bit challenging because we're measuring. Is that not the case?
Oh no, it's just I am working with validating DCX against perfusion and MRI.
Okay.
And, we also have TSP measurements and patients and stuff. So there's a drawback with everyone.
Yeah.
I'm just curious what your thoughts are.
Yeah. So right. I mean, I think those are good directions you're pursuing. There is no ideal validation, right? So kind of as I said, there's no real technique that measures cerebral blood flow. I mean, even some of these MR techniques, they're measuring plasma essentially and correcting for hematocrit, and you're measuring red blood cells. So even at that basic level, like the core CBF techniques are not really measuring CBF. So I think it will be a question of a body of evidence that accumulates over time, and then people will start to accept it, and soon they won't even care, has this been validated? The measurement is just useful in its own right. But I think maybe we're not quite there yet.
Kind of like how perfusion MRI is validated to a standard, it's very useful. It's also super noisy. And so, but people don't really talk about that. It's just accepted.
I haven't read your latest optical paper. What's your fastest camera now, or how fast are you go? Like, what's the?
This one is at 333 kilohertz.
Okay. So you're really just catching the tail end still, the curves, or?
So there is some decorrelation over the. So the
Yeah.
question is about the sampling of the autocorrelation function, which I think we should also mention is a limitation of this technique, because DCS with a photon counting, you can achieve kind of arbitrarily fine sampling of these fluctuations, whereas if you have a camera, you're kind of limited by the sampling rate that the camera manufacturer provides to you. That is a limitation, and that's one of the reasons we're not pushing to further source detector separation. It turns out with the time of flight filtered approach and with filtering, the decay rate goes down a little, so it's a slower decay rate when you use time of flight filtering for the same level of brain specificity. So it does help a little bit, but that is a concern that will need to be worked out.
I'm wondering, you talked about OCT and then this diffuse optic interferometric approach. I mean, what are your thoughts about potentially combining some of the OCT information together with a scal blood flow, for example? Is that something you thought about?
Yeah, I thought we definitely. I mean, I think the scales are just so different. It's hard to get useful information. So I mean, ideally, you'd want to measure the scalp with an exact same weighting as what your deep measurement with diffuse light is giving you, and that's just very difficult to do with any technique that I know of. But having the ability to isolate early times of flight is actually pretty good because you're using the same source and detector. You're just getting early times of flight and late times of flight, and you can use them to regress out the superficial tissues.
Great. Well, thank you so much. This is a wonderful talk. Let's give our speaker
Thank you.